أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. Dear students, today my topic of discussion is about cell and using a light microscope, and we will compare animal cell and plant cell. And at the end of the lesson, you will be able to understand what is cell, to understand the difference between unicellular and multicellular organisms, and to understand how to use a light microscope, and to understand the similarities
ये
and
लिखना है
no differences between animal cells and plant cells. Okay, so first of all, what is a cell? Okay, the cell is the basic structure and functional unit of all known organisms. Cells are often called the building block of life. All the living organisms are made up of cells. So that is why it is so called building block. Okay, because without a cell, a living organism will not exist. Cell is the basic
अब
structure
ऐसे कैसे हो रहा है
and functional unit of all known organisms. Okay, it will perform all the basic functions of an organism, and without a cell, the survival of an organism is quite impossible. Now come towards cells. All living things are made up of one or more cells. Some of them were so-called unicellular because their bodies were made up of only one cell, and some of them are so-called multicellular because their bodies are made up of millions of cells. Some living things such as bacteria exist as a singular cell. For example, bacteria, amoeba, chlamydomonas, paramecium, all of them were so-called unicellular because their bodies were made up of only one cell. They are so-called unicellular organisms. Most animals and plants, however, are made up of millions of cells. Okay, millions of cells, they were made up of millions of cells, and that is why it is so called multicellular organisms. For example, we human beings are multicellular organisms. For example, god, sheep, buffaloes, and some big plants as well. So their bodies were made up of millions of cells. They were so called multicellular organisms. So let me revise the things again for you, that those animals whose bodies were made it from only one cell, they were so-called unicellular organisms. For example, amoeba, you can see here in this picture, this is amoeba, okay? You can see here in this picture, this is so-called amoeba. Their body is made up of only one cell. And other example of unicellular organisms are Chlamydomonas, Euglena, Paramecium, bacteria, all of them are so-called unicellular organisms. In spite of unicellular organisms, there are animals whose bodies were made up of millions of cells, and they are so-called multicellular organisms. And they will be numerous in number. For example, human being, goat, sheep, buffaloes, some big trees. Okay, so they were so-called multicellular organisms. We can't see cells. Okay, so that is why it is so called microscopic. Okay, cells are microscopic. What does it mean cells are microscopic? Because we are not able to see them through our naked eyes. So all those things which cannot be able to seen by our naked eyes, we call them microscope. Okay, and those things which we can see with the help of our eyes, we can say them that they were so-called macroscopic organisms. This means that they are very tiny and can only be seen with the help of a microscope. When you look at an object through a microscope, if you can see an object through a microscope, you can see here at the bottom of your screen, you can see here, this is also called microscope. Okay, the object appears larger because the microscope magnifies what you can see. What is the purpose of the microscope? Microscope magnifies. It will increase the size of the object. It can increase the size of the image of the object. Okay, and as a result, we can see them easily through microscope. Those organisms which cannot be able, which we cannot see with the help of our eyes, we can see them through microscope, because microscope is a device with the help of which we can see microorganisms. The most common type of microscope is called light microscope, because we use that microscope with the help of light. Light will pass in from the specimen. So that is why it's so-called a light microscope, because it uses light to illuminate the object. The diagram shows a typical light microscope. You can see here in this edge of your corner. So this is what so-called light microscope. Okay, we can say this microscope is as a light microscope. Now, this is what's so-called a microscope, and we will explain the different parts of this microscope briefly. Okay, so you should come on page 19, and you can see here this diagram of microscope. I will start from the very beginning. Okay, at the top of the microscope. So at the top of the microscope, there is a lens, that is what so-called eyepiece. Okay, this is a lens. This lens is so-called eyepiece lens. Okay, and the function of this lens is to, the eyepiece contains the lens which the user look through to see the magnified specimen. Okay, you can use this lens, and with the help of this lens, you can place your eye in front of them. Okay, and as a result, you can see the specimen. Okay, now come towards the lower two knobs. These are so-called focusing knobs. Okay, these two knobs are so-called focusing knobs. With the help of these two knobs, you can place your eyepiece and your object pieces towards the specimen. With the help of these two knobs, you can adjust the both the lenses. Okay, so sharpen the focus quality of the image. We use focusing knobs. Now, beside these focusing knobs, you can see here these are three other lenses, and these three lenses are so-called objective lenses. Okay, they were so-called objective lenses. Why it is so-called objective lenses? Because these lenses were in front of the object. Okay, they were in front of the object, so that is why it is so-called objective lenses. And the function of the objective lenses is, the objective lenses combines with the eyepiece lens to increase magnification level. Okay, it can increase the magnification level. It can magnify the image, or it can magnify the image of the specimen that were placed there on a stage. Okay, lower you can see here these are so-called clips. Okay, this is a clip, and it is used to hold the slide firmly in place. Okay, if you want to fix the slide, so you can use the stage clips. Okay, with the help of the stage clips, you can hold the slide firmly there. Okay, if you fix them with the help of the clips, then the slide will be unable to move, and you can easily examine the specimen there. Below that, you can see here, this is what's so-called stage. Okay, this is the platform where you can place the slide. Okay, you can place the specimen here on the stage. So this is what's so-called stage. Below this is so-called diaphragm. Diaphragm, remember one thing, that diaphragm is a thing that can be transparent, with the help of which light can be passing through them. Okay, so that is what so-called diaphragm. And the function of the diaphragm to control the amount of light or reaching the specimen. It controls the amount of light that can reach the specimen. So that is what so-called diaphragm. And below you can see here, this is so-called mirror. And the function of this mirror is to reflect light through the specimen. Okay, it reflects the light, and that light will passing from the specimen, and after passing from the specimen, it will enters to the object piece, and to the object piece it will be passing there to the eyepiece, and as a result you can see the specimen there. So this is all about the different parts of the microscope. I hope so that this thing will be quite clear to you all. Okay, there are so many parts of the microscope. The first one is so-called the eyepiece. Okay, there are some focusing knobs from which you can adjust the lenses towards the specimen, and through beside them there are some lenses that are so-called objective lenses. Okay, with the help of them you can easily see the objects, and below that there is a stage with stage clips, with the help of which you can place the specimen to be examined. Below the stage you can see there is a diaphragm which can control the amount of light that will pass in from the specimen. Okay, and below that you can see that there is a mirror, and the function of the mirror is to focus the light to the specimen, or to focus the light through the specimen that can pass in from the specimen, and you can easily examine the specimen there. So all of these are the parts of the microscope. Okay, that is what's so called a light microscope, because with the help of light we can examine the different things. Okay, with the help of them we can examine different things, and as a result we can magnify the images and we can be able to easily see the things that can be placed in the on the slides. Now, using a light microscope, so the object is put on a glass microscope slide and usually covered with a cover slip. First of all, you should prepare a slide for yourself. Okay, you can place the microorganism on a slide and you can fix them with the help of the cover slip. Okay, and then the slide is placed on the stage. You place the slide on the stage of the microscope, okay, and held in place with the stage clips. You can firmly fix the slide there with the help of the stage clips. Then the hole in the stage allows light to pass through from the source below. Okay, this mirror will reflect the light that can be that can reflect the light that can pass through the specimen. Okay, the diaphragm can be adjusted to allow more or less light to pass through the hole. Okay, this is the diaphragm. You can see here, this below the stage, you can see here, this is the diaphragm, and it can be used there to control the amount of light that can be entering, that can reflect it from the mirror and can pass in from the specimen. Light shines through the object and into the objective lenses. Light passes from the objective lenses through the eyepiece lens and into your eye. Then this light will pass in from the specimen. It will enter through the object lens, and the object lens will be focusing it to the eyepiece. And with the help of the eyepiece, you can be easily see that object. Okay. The focus knob adjusts the distance between the stage and the objective lenses to create a clear image. Okay, so this is the use of a microscope. Okay, first of all, you can prepare a slide for yourself. Then you can fix the slide with the help of the stage clips. Then you should adjust the light that can pass in from the slide, okay, with the help of the diaphragm. And then that light will pass in from the slide. It will enter to the object piece. The object piece will focus that on to the eyepiece, and through eyepiece you can easily able to see the object that can be examined there through microscope. So this is all about a microscope. Light microscope usually have more than one objective lenses. Okay, you can see here there are in this figure you can see here there are three objective lenses. Okay, these are three objective lenses: one, two, and three. These are three objective lenses. So light microscope usually have more than one objective lenses. These are arranged on a turret which can be rotated so that you can use lenses with different magnification. Each and every object lens have different magnification level. For example, some have x5, it means it will magnify the image 5 times. Some have x10 magnification, that it can magnify the image 10 times. And some have x20, it magnify the image 20 times, and x40 means it can magnify the image 40 times. Okay, so the eyepiece lens is usually x10. The eyepiece usually it magnifies the image 10 times. If the eyepiece magnifies is x10 and the object lens magnifies x20, then the total magnification is 10 multiplied by 20, and it will 200 times magnifies the image. Okay, if you have the eyepiece magnification is 10 and object piece magnification is 20, so you should multiply 10 with 20 and it will give you 200. It means that the image will be magnified 200 times of its original size. Okay, so this is the formula from which we can magnify the things. Okay, animal cells and plant cells. Here we are here to compare different cells, that is from animals and from plant cells. So animals and plant cells have similar job to do. They take in food, releases energy, get rid of waste, grow and reproduce. Their structures, however, are not the same. Different cells have different function and different structures. For example, the cells are there in your cheek will be quite different from that of your skin, and the cells of the skin will be quite different from that of your eyes. and the cells of your eyes will be quite different from that of your tongue, and the cells of your tongue will be quite different from that of your legs. So different organs have different types of cells are there. Okay. Similarly, the cells of the leaves will be quite different from that of the cells of the stem, and the cells of the stem will be quite different from that of the cells of the roots. Okay, so different organs have different types of cells, and we will compare them accordingly. You can see here, this is an animal cell. Okay, at your left, you can see here, this is an animal cell, and this is a plant cell. There are some similarities, some similar things were here, okay, and some different things are also here. For example, both the cells, both the animal cells and plant cell have cell membrane, okay. The outer layer of the cell, that is so-called cell membrane, and you can see here, both the cells have a cytoplasm. Cytoplasm is a liquid in which all the organisms will be floating there. Okay, all the organisms of the cell will be floating through cytoplasm. Okay, so cytoplasm is a jelly-like structure, and its function is that all the organisms or the organs of the cell will floating in cytoplasm. And you can see here that both the cells, both the animal cells and plant cells have nucleus. Okay, so both the cells have nucleus. These are so-called similarities. Okay, we can say that these are common in all. Okay, so they were so-called similarities. Now there are some differences between the animal cells and plant cells. The plant cells have a cell wall, okay, but the animal cells do not have any cell wall. That's why the skin or the surface of the plant will be quite harder as compared to the animal cell. Okay, if you touch an animal cell, so that will be soft. But you can, if you touch the skin, if you touch the skin of the plant, so you feel that the skin of the plant is quite harder. Okay, that will be quite harder. So that is because of the cell wall. Okay, that is because of the cell wall. And the plant cells have a large vacuole. Okay, in spite of large vacuole, in animal cells they have some smaller vacuoles are there. And the function of the vacuole is to collect all the waste materials from the cell, and it will be removed from the cell. Okay, also, in plant cells, you can see here, these are some pigments. These green pigments are here. And the purpose of these green pigments, these are so-called chloroplasts. And the function of the chloroplast is to prepare food for the plant. Okay. So these green pigments are so-called chloroplasts. And the function of these green chloroplasts is to prepare food for the plant. Animal cells versus plant cells in detail. Now we can explain these things in details. So animals and plant cells have three similarities: a nucleus, which is the control center of the cells. The nucleus contains the information which controls everything that goes on in the cells. Okay. So first of all, what is nucleus? Nucleus is the control center of the cell, where either that will be animal cell or plant cell. So the nucleus is the king of the cell. Okay, that controls all the activities of the cell. Okay, nucleus is the control center of the cell. With the help of the nucleus, all the activities there in cell will be controlled. The nucleus contain information which control everything that goes on in the cell. Now come toward the second thing. The second thing is the cytoplasm. So the cytoplasm is all the living matter except for the nucleus. Okay, the cytoplasm is all the living matter except for the nucleus. It is a jelly-like substance which is float, can flow. Earlier I told you that cytoplasm is a jelly-like structure in which all the organelles of the cell floating there. Okay, it will provide some space, some liquid to the organelles that can float through there. So that is what's so called cytoplasm. Now come towards the cell membrane. It is the outer layer of the cell. Okay, the cell membrane is the thin skin around the outside of the cell. It is the outer layer of the cells. It prevents the cell's content from escaping. Okay, it stops the organisms that can passing from the cell. If there is no cell membrane in the cell, then the organisms of the cell will be quite easily escaping from the cells. So it will restrict the organelles inside the cell. Okay, so these are so called the boundaries They were so-called boundaries. Okay, cell membranes is so-called the boundary of the cell, which limited the cell, and it would allow the organisms to stay inside the cell. But it does not allow substances like water and other small molecules to cross in and out of the cell. It will allow only smaller objects, smaller molecules to get in and to get out of the cell. Okay, so the function of the cell membrane is that it will restrict the organisms inside the cell, and it will allow only water and minerals that are required for the survival of the cells. It will allow only water and minerals to get in to the cell, and it will allow the waste materials that were collected by the vacuole, okay, and it will be removed from the cell, okay, at certain time. So this is the function of the cell membrane. Come towards the plant cell. In addition to these, plant cells have three other important features that these features are not there in the animal cells. The cell wall is a rigid foam coating around the outside of the cell membrane. It helps the plant's cells keep its shape. Okay, it can make shape of the plant. It gives a shape to the plant cell because it is hard, it is firm, it is rigid. Okay, so that's why it gives a specific shape to the cell. Chloroplast contains a green chemical called chlorophyll. Okay, you can see here these green pigments. They were so called. These green pigments are so called chloroplast. Okay, and what is the function of the chloroplast? It contains chlorophyll. Okay, and this is the chemical which enable green plants to use the sun energy to make food during photosynthesis. These uh, these pigments are responsible for making food for the plant during a process called photosynthesis, where uh, photosynthesis is a process through which plants make their own food. Okay, so it's a process by which plants make their own food. That is also called photosynthesis. The large vacuole is a large space in the middle of the cells. It is filled with a fluid called cell. Okay, this is the vacuole. Okay, and it is always located at the center of the cell. And the function of the vacuole is to collect all the waste materials. Okay, and it have a liquid that is also called cell. Okay, now complete the table. Nucleus, they have dense nucleus. They were dense round structure. Okay, and what it does, so it is the control center of the cell. Now cytoplasm is fluid substances, and everything floats in this, so that is what's called cytoplasm. Now an interesting information for you: an electron microscope achieves a much higher magnification than a light microscope. It means that electron microscope is a microscope which will give millions times more magnified image as compared to the light microscope. This is because it passes a stream of electrons through the object instead of light. Okay, so a stream of electrons passing from the object, and it will give a millions times magnified image as compared to that of the light microscope. Time to review. So all living things are made up of one or more cells. Some living things, such as bacteria or unicellular organisms, plants and animals, are called multicellular organisms. The next thing is plant and animal cells have three common organelles, that is so-called nucleus, cytoplasm, and cell membrane. They were common in both the plant cell and animal cells. And the third thing, that is cell wall, chloroplast, and larvacule are present in plant cells but absent in animal cells. So this is all about today's lecture. Okay, and you can do this assignment by yourself on page 20 of your book. Okay, so you can do this by yourself. And now it's time for questioning. So if you have any question, you should ask, please.